Welcome back to the deep dive. You sent us some really fascinating material this week, and we are going straight to the cutting edge of longevity science. Today, we're attempting something pretty ambitious, trying to synthesize the molecular blueprint of biological aging with the AI-driven tech that's actually designed to disrupt it.
Yeah, it's an incredible convergence, isn't it? So to guide you, we've got two really foundational sources. The first is this extraordinary piece of research. They're calling it an aging atlas. It used, get this, over 15,000 human methylomes across 17 different tissues. So we're not just looking at how your blood is aging anymore. This gives us a really deep tissue-specific map of our molecular clock.
Okay, molecular map.
And the second source.
The second source zooms right in on therapeutic intervention, specifically tackling that notorious NLRP3 inflammasome. And this analysis really showcases how a new AI-enabled workflow, it's called Legion, is being used to explore, well, millions of potential molecules to finally try and switch off that chronic inflammation. Right, so the mission today is map the aging process and see how we're hunting for the fixes.
Exactly. We want to give you a clear view of both the foundational science like the detailed architecture of aging from the atlas, and then the most promising paths forward in finding those master switch drugs that could actually maybe roll back the clock a bit. Let's start with the map itself, that huge DNA methylation atlas.
Okay, let's dive into the scale here, because 15,000 samples across 17 tissues sounds massive. We often hear about epigenetics, DNA methylation, these chemical tags switching genes on or off. Why is looking at 17 tissues instead of just blood like usual such a big deal?
Well, the key realization is that aging isn't monolithic, right? Your liver, your heart, your brain, they all age under different stresses, different regulatory pathways. By pulling together 15,000 samples across all these tissues, we move past just a general age estimate. We start to understand how specific organs are actually changing or maybe failing. The scale lets us see the common themes, sure, but also those really crucial tissue-specific differences.
And they didn't just use one measure, right? The source mentioned 3 distinct ways they looked at aging through methylation.
That's right. Think of it like a car odometer, maybe, but one tracking 3 different kinds of wear and tear. First, you've got the DMPs. That stands for differentially methylated positions. These are the predictable sort of directional shifts. If the biological clock says you should lose, say, 5 methyl groups at this spot every decade, and you pretty much do, that's a DMP. They're the consistent biomarkers, the ones that reliably tick along with age. Kind of the official clock reading.
Okay, so DMPs are the expected changes, the steady ticking. What about the unexpected stuff, the glitches?
That brings us to the second dimension. VMPs, or variably methylated positions. These capture the more stochastic component, the random damage, you could say. Think of it as the accumulated unique noise or wear and tear that happens over a lifetime. It reflects the increasing variability between individuals, why your aging pattern might look a bit different from mine, based on lifestyle, environment, that sort of thing.
Okay, so DMPs are predictable aging, VMPs are the unpredictable individual noise, and the third metric, Shannon entropy, sounds complex.
It sort of quantifies the overall epigenetic disorder. Imagine your genome is like a perfectly organized library when you're young. Everything's in its place. As you age, maybe books start getting put back randomly, titles get smudged, things aren't where they should be. High Shannon entropy means high disorder unpredictability across the whole system. It's basically a measure of how messy and unstable the entire molecular setup has become.
That's actually a really helpful way to picture it. The predictable breakdown, the random wear, and just the total messiness. So when they map those predictable changes, the DMPs, across the 17 tissues, what did they find? Did some organs stand out as aging faster or differently?
Yeah, there's extreme tissue specificity, really striking. Brain, liver, and lungs showed the largest numbers of these predictable changes, the DMPs. The brain, for example, had over 180,000 DMPs. That's a massive signal. It tells us the brain undergoes really profound, predictable epigenetic remodeling as we live our lives.
Wow, but then some organs must be more stable, right? Or maybe they just age in a completely different way.
Exactly. Tissues like the pancreas, the retina, even the prostate they showed minimal, or in some cases like no significant DMPs at all. Now, this doesn't necessarily mean they aren't aging, but it suggests their epigenetic landscape might be inherently more stable, perhaps more buffered against these purely chronological changes. It raises a huge question. Why are their regulatory systems so resistant to this kind of drift?
Interesting. Okay, what about the direction of change? Are we generally gaining methyl tags or losing them as we get older?
The general trend, across most tissues anyway, was towards age-associated hypermethylation. so gaining methyl groups. This often happens in regions that were previously unmethylated or open in younger people. So it suggests a kind of progressive chromatin closure, like the cell is slowly locking down or silencing genes.
Which makes intuitive sense for declining function, right? Things get locked up, silenced, less active.
That's the general thinking, yeah. That's the overall pattern. But, the exceptions are always where it gets interesting. Skeletal muscle and lung tissues actually showed heightened hypomethylation, a loss of methyl groups. So in these cases, key regulatory regions might be opening up when they shouldn't be, maybe leading to dysfunction. It clearly points to tissue-specific aging pathways needing potentially different kinds of intervention.
And the brain.
Yeah.
Which had the most changes overall? Did it just follow the hypermethylation trend too?
No, the brain was distinct again. It showed a much more balanced mix of DMPs across all methylation levels, low, medium, and high. It suggests its aging isn't just about progressive silencing. It seems to be a more sophisticated, maybe more buffered landscape, allowing for both silencing and activation in a really complex regulatory dance.
Okay, so we've got this incredibly detailed map now. Does it point towards any, universal targets? Did they find a specific gene family that seems central to this whole process across multiple tissues?
They did, yeah. They used network analysis to sort of connect the dots, and it pointed pretty directly to the protocadherin gamma family, or PCDHG. These genes popped up as key drivers, or hubs, not just in the overall pan-tissue analysis, but specifically in adipose tissue, blood, brain, and skeletal muscle. The thinking is they might be critical for maintaining, structural integrity and signaling balance in these really key tissues.
Right, so that gives us a potential molecular address to target. But the analysis also found evidence for something else, right? A pathway that's actually resilient against aging, which seems just as important. Tell us about the NAD+ connection.
Oh, this was one of the most compelling findings, I think. The researchers did this in silico robustness analysis. Basically, they simulated stress on the system computationally to see which epigenetic modules fell apart and which ones held strong. As you'd expect, most modules were pretty fragile.
They broke under pressure.
Yeah, they broke. But they found one incredibly resilient module, the survivor, you could say. And the survivor module was significantly enriched for pathways involved in NAD plus salvage metabolism. This provides really strong independent epigenetic evidence supporting the idea that maintaining NAD plus levels isn't just, a passing fad in longevity circles. It seems to be a core resilient pathway crucial for keeping cellular balance across multiple tissues as we age. It kind of validates years of research in one neat finding from this huge data set.
Okay, that's the molecular architecture laid out really clearly. We know where aging is predictable with DMPs, where it gets noisy with VMPs, and we have potential structural targets like PCDHG and maybe reinforcing agents like NAD+1. Now let's pivot smoothly to the active hunt for interventions, specifically targeting what you called the ultimate master switch, the NLRP3 inflammasome.
Yeah, it's a perfect transition because if aging involves this sort of growing disorder and chaos, NLRP3 is often the trigger that turns that chaos into actual disease. We often call it the Swiss Army knife of inflammaging, that chronic, low-grade, sterile inflammation that seems to drive almost every major age-related disease. Neurodegeneration like Alzheimer's and Parkinson's, atherosclerosis, fatty liver disease, or NIH, even kidney disease. So the idea is, if you can shut down NLRP3, you might mitigate a whole host of risks all at once.
The theory sounds great, but now we're actually seeing it play out in the clinic.
Yeah.
Let's talk about those pretty striking phase 2 results for Venus's oral NLRP3 inhibitor, VTX3232, particularly in cardiometabolic health.
The results were genuinely phenomenal, especially in this group of patients with obesity and high cardiovascular risk. The main outcome was a nearly 78% reduction in high-sensitivity C-reactive protein, or hsCRP, levels after just 12 weeks. And hsCRP is a really well-established marker for systemic inflammation and cardiovascular risk.
78% is a huge drop, but maybe explain for the listener, why should they care about lowering hsCRP? What does that actually mean for their health?
Right. Good question. A high hsCRP level basically means your body's inflammatory system is running hot, kind of simmering all the time. This increases your risk for things like heart attack and stroke. It shows chronic inflammation is active. So cutting that by nearly 80% suggests we're dramatically cooling down that baseline inflammatory risk. We're potentially moving someone from a high-risk inflammatory state towards a much calmer, healthier internal environment.
And here's what jumped out at me from the source material. The drug didn't cause weight loss. Why is that specific point so significant?
Because it proves that NLRP3 inhibition targets the residual inflammatory risk directly. Think about it. Many modern obesity treatments, like the GLP-1 drugs everyone talks about, work by reducing appetite and improving metabolism. They often lower inflammation too, but largely as a side effect of the weight loss. VTX3232 showed it could slash inflammation dramatically,
Oh.
independent of that weight loss pathway. It's hitting the inflammation itself.
So we should think of these as potentially complementary treatments, not necessarily competing ones.
Exactly. The data strongly suggested that adding the NLRP3 inhibitor on top of standard treatments, including GLP-1s, gave additional reductions in inflammatory markers like hsCRP, IL-6, even LPA. This really bolsters the idea of a potential new therapeutic triad for tackling aging and cardiometabolic risk. You'd have statins managing lipids, GLP-1s handling metabolism and weight, and then NLRP3 inhibitors specifically targeting that underlying inflammation.
And the momentum isn't just in heart health, right? We're also seeing big pushes in neurodegeneration, specifically Parkinson's, where inflammation is known to be a major player.
That's right. Other companies are in the mix too. For example, Nodthera's trial with their drug NT0796 showed it reduced inflammatory biomarkers like IL-1 and IL-6 in patients' cerebrospinal fluid. And crucially, it also seemed to reduce markers of actual neuron damage, like NFL. That hints maybe at protecting the neurons, not just managing inflammation symptoms.
And Ventyx's drug, VTX3232, also showed promise in Parkinson's.
Yes. Early open-label data suggested it could cross the blood-brain barrier effectively, which is key. It led to significant reductions in inflammatory cytokines in the brain, and perhaps most excitingly, showed a statistically significant improvement in both motor and non-motor symptoms on the standard Parkinson scale, the MDS-UPDRS. Still early days, but very promising.
Okay, so this is clearly why there's such intense focus on finding the best NLRP3 inhibitors. If you have a target this valuable, potentially modifying diseases like Parkinson's, you need the best tools to find the perfect drug. Which brings us back to the AI.
Precisely. The challenge is just
Yeah.
mind-boggling. Chemical space, the theoretical universe of all possible drug-like molecules, is estimated to contain up to 10 to the power of 60 structures. That's astronomical. How do you even begin to search that?
You can't do it with traditional lab screening, that's for sure.
Exactly. You need systems like this Legion workflow they described. It stands for latent enumeration, generation, integration, optimization, and navigation. Quite a mouthful. But essentially, it integrates specialized generative AI engines, like things called Zero PC and MolGrow, with AI-guided screening platforms like Chemistry 42.
So instead of physically making and testing billions of compounds, Legion uses AI to imagine or generate molecules in virtual space first.
That's the core idea. It's designed to intelligently navigate what they call fraction 2, the parts of chemical space that are synthetically accessible, meaning we could make them, but they just haven't been explored yet. It uses deep learning to figure out the geometric shapes and electronic properties needed to actually bind effectively to a complex target like NLRP3.
And using NLRP3 as its target, what kind of scale did this Legion system actually manage?
It was pretty impressive. The workflow successfully identified over 34,000 unique chemical scaffolds, sort of like basic molecular skeletons that fit their 3D binding ideas for NLRP3. Then, using these scaffolds as starting points, the AI generated approximately 110 million distinct molecular structures for further virtual screening. And crucially, it focused on ones that look structurally plausible to actually synthesize later.
110 million generated molecules. Wow. But was this AI approach genuinely more efficient than just, say, randomly generating and testing millions?
Significantly more efficient, according to the source. The AI-guided 2D generative chemistry methods showed a really remarkable 58.8% hit rate in the follow-up 3D virtual screening step. Compare that to more traditional combinatorial explosion methods, which typically yield hit rates down in the 8% to 26% range. It shows the AI isn't just generating random junk, it's generating structures that are highly relevant to the specific 3D shape of the target. That saves immense time and resources filtering out irrelevant compounds.
Okay, let's try and tie these two huge themes together then. We've seen this incredibly detailed granular map of biological aging emerging from the 17-tissue atlas, and we've seen this accelerated AI-powered hunt for drugs targeting that key inflammation master switch, NLRP3.
Yeah, the picture emerging is pretty clear, I think. We now possess a detailed, tissue-specific molecular blueprint of how we age. We know the process involves both these predictable structural shifts, like we saw with the PCDHG genes, and the resilience of certain core metabolic pathways, particularly NAD plus salvage. And maybe most importantly, the clinical evidence is now confirming that targeting the downstream consequences, like the inflammation driven by NLRP3, is already yielding really dramatic risk-reducing results in humans.
It really feels like we're shifting gears, moving from just observing aging to genuinely intervening, and at a speed that, frankly, would have seemed like science fiction just a decade ago.
Absolutely. And that speed is being driven by technologies like Legion. So here's maybe a final provocative thought for you, connecting our two sources directly. If AI can now efficiently navigate vast chemical space to design drugs for a master switch like NLRP3, how long will it be until we see AI-designed drugs created specifically to target and correct those tissue-specific epigenetic drivers we talked about earlier? Things like the unique hypomethylation patterns in the aging lung, or boosting that resilient NAD plus metabolism where it's needed, or maybe even subtly influencing those PCDHG structural changes. That level of truly tailored, maybe even preventative molecular medicine based on your specific aging map. That's what this convergence promises, and that's the next frontier we'll definitely be keeping an eye on.